Hello everyone, you're watching Dr. Vipin Kumar, and today in the series of the plant physiology, this is actually first series. Here I am going to tell you about the 10 most frequently asked questions in plant physiology. So let's start the first question. The first question is saying that the enzyme for the conversion of ammonia to amino acid is. So here our answer will be C, glutamine synthetase. Let's start some more about the glutamine synthetase. So here I have given some explanation of the glutamine synthetase. Actually, this enzyme is having the important essential role in metabolism of nitrogen by catalyzing the condensation of glutamate and ammonia to form glutamine. Here we can see that here glutamate in the presence of ATP, because it is an energy consuming process, and ammonia, it will form the glutamine. And actually this glutamine is used as a base material for the synthesis of the protein, or we can say the metabolism of the nitrogen. So this is actually the role of glutamine synthetase. Let's move to the another question. Another question is saying that important function of leg hemoglobin in root nodule is. So here our answer will be oxygen regulation. As we know that the oxygen regulation in leguminous plant is very important for the active functioning of our nodule. Here I am going to show you some figure of root nodule. Here you can see that this is actually the root of a leguminous plant, and these root are covered with nodules, and this is actually called bacteroid, means nodule plus bacteria. This is actually called the bacteroid, and the activeness of this nodule are determined by the color of the nodule. If suppose this is look like pinkish color, it means that it is the active root nodule, means nitrogen fixation is going on. So here are some given some explanation about the lag hemoglobin. So here we can see that lag hemoglobin is a nitrogen or oxygen carrier and hemoprotein found in the nitrogen fixing root nodule of leguminous plant. And lag hemoglobin has a very, what we can say, that close chemical and structural similarities to the hemoglobin that is in our body or in mammals body, and that looks in red color. And actually hemoglobin also called as a holoprotein. It means that protein plus hem, and hem means iron. It is act as a cofactor, and it is widely believed that the product this is the product of both plant as well as the bacterium. It is widely believed that what is this holoprotein? Actually this is the product of both plant as well as bacterium. Here we can see that in which the apoprotein that is produced by the plant and the hem that is actually produced by the bacteria. It is widely believed that this is the actually come from the both our plants part as well as the from bacterium plant. So this protein is actually called holoprotein, and or sometimes we can say that it is the lag hemoglobin. Okay, let's move to another question. The third question is saying that the externally thin strand of cytoplasm is termed as. So here our answer will be D, Plasmodesmata. So let's move for some more explanation about the Plasmodesmata. Here we can see that the structure of the Plasmodesmata. Plasmodesmata will be very thin membrane here, and the role of this Plasmodesmata is to make a connection between the different cell. It will make a communication. So here I have already given some explanation about this Plasmodesmata. Actually if you see the Plasmodesmata, this is a microscopic channel. Here you can see which traverse the cell wall, that will be traversed through the cell wall here, okay, of the plant, and some algal cell, and enabling the transport and communication between them. So it will go through the cell wall between the two cell of the plant, and it will make a transport of the transport as well as the communication. Here more explanation are given like that. Here you can see that neighboring plant cell are therefore separated by the pair of cell wall and the intervening middle lamella, forming an extracellular domain known as apoplast. So side of that, if you see that, that will be separated by the another structure here, looking like the yellow in color, that is actually called the apoplast. And what is the apoplast? Here if you see, this is the apoplast, is means the movement will be occur, the pathway will be occur through the cell wall. And if we are talking about a cymoplastic pathway, it means that that will traverse or move through the cytoplasm. Okay, I hope you got the clarity in between the apoplastic pathway as well as the symplastic pathway. Symplastic pathway means through the cytoplasm, and apoplastic pathway, it means through the cell wall. So there will be some structure side of this, what we can say, our plasmodesmata, and that will be the apoplast. Okay, and although the cell wall are permeable to small soluble protein and other solutes, plasmodesmata enable the direct and regulated symplastic transport of the substance between the cell. I have already told that the role of this plasmodesmata is to make a communication as well as the supplying some material in between the cell. So these were some of the function of the plasmodesmata. I hope you got some clarity and clarification about the plasmodesmata. Let's move to another question. Another question is saying that in plant, the genetic material present in, actually genetic material, if I am talking about genetic material, it means that DNA and RNA. Okay, and these are genetic material present in, our answer will be here nucleus, chloroplast and mitochondria, means D is the right answer. So let's move some explanation about this genetic material. So what is the genetic material? Genetic material, if you see that the genetic material here are given that the genetic material of cell or an organism refer to those material found in nucleus, mitochondria and cytoplasm. This was actually the questions in our previous question, and the answer is the nucleus, mitochondria and cytoplasm. And the role of this genetic material is to determine the structure, nature of cell substances. Okay, so on the basis of genetic material, the organism will display their proper structure and nature of the substances, and it will have the property of self propagating and variation. This genetic material will have a capability to propagate itself as well as will make a variation in the population. Okay, so that's why the individual looks different from the other. So this is actually the role of the genetic material. So let's move to the another question. The fifth number question, the main function of endoplasmic reticulum is. So from here our answer will be B, protein synthesis. And here the structure of endoplasmic reticulum we can see here. This is the structure. So endoplasmic reticulum will be two type: smooth endoplasmic reticulum and rough endoplasmic reticulum. So actually this is the two type of the endoplasmic reticulum are found. And apart from that, you can see also there is the cisternal space here, and this is actually this rough type of structure, those are visible here. This is actually the cisternae. Okay, and this is the nuclear envelope. Okay, so these are the some of the structure of the endoplasmic reticulum. So let's move some information about the endoplasmic reticulum. Here I am giving. Endoplasmic reticulum is a type of organelle found in eukaryotic cell that form a interconnected network of flattened and membraned enclosed sac or tube-like structure known as cistern. So actually it will found mainly in the eukaryotic cell, and it make a interconnection of the network in between of flattened and membrane enclosed sac-like structure. What I have shown to you, there are some structure of the cisternae, so it will make a interconnection of the network among that. And the membrane of endoplasmic reticulum are continuous with the outer nuclear membrane. The endoplasmic reticulum occur in most type of eukaryotic cell, but is absent in red blood cell and spermatozoa. So this is a very important fact you can write down. It may be asked somewhere else. And other fact here are we can see that there are two type of endoplasmic reticulum. I have already explained to you. One is the rough endoplasmic reticulum, other is the rough endoplasmic and smooth. There is the two type. And if you see that above the rough endoplasmic reticulum, there will be some studded structure studded with the ribosome, and that is actually the site of protein synthesis. So this was actually the questions in our previous question. So that in that question, the question was asking what is the actual role of endoplasmic reticulum. So from here you can see this is actually the answer of that. The rough endoplasmic reticulum is especially prominent in cells such as a hepatocytes. So this will be your very important fact. You can write down. This may be asked in your any exam. That is actually seventh number of the question. Seventh number of question is saying that Golgi body was discovered in 1898 by. So simply this answer will be B, Camillo Golgi. So let's move further. Some more explanation about that. So the explanation of this actually Golgi apparatus. Golgi apparatus is also known as the Golgi complex, Golgi body, or simply sometimes we can say that Golgi, and is an organelle found in the most of the eukaryotic cell. And this was actually identified in 1897 by an Italian scientist, that's name is Camillo Golgi. And so by the name of that, these organelles are given the name of Golgi apparatus in 1898. And part of the endomembrane system in the cytoplasm, the Golgi apparatus packages protein into membrane bounded vessels inside the cell before the vessels are sent to the other destination. So here we can say that this having an important role for the package. It will make a package of the protein in a vessel's form. Before that the vessel will be transported to the destination. Suppose suppose from this place to this place, A to B, suppose the protein transportation are required. So here it will make a sac or vessels, and in that it will store the protein. So that vessels will transport here from here to here, and this is actually the function of the Golgi apparatus. So we can understand that what is the role of Golgi apparatus. It will make a transportation of the protein from one place to other, means our destined place. Let's move to another question. This is the 8th number of the question. 8th number is saying that RNA synthesis in nucleus participate in protein synthesis in. So here our answer will be cytosol. So what is the actually this is? Let's move. Actually, this is the this figure is actually the this is actually the cytoplasm, and actually the cytoplasm will comprise both cytosol as well as the organelles found inside the membrane of the cell except to the nucleus. Okay, this will be our cytoplasm. So cytosol is the site where the RNA synthesis will be occur, and the cytosol is actually the in the form of liquid matrix that will be found around the organelles inside the cell membrane. That is actually the cytosol, and that will have the component of the salt, water, organic molecules, and cytoskeleton filament. Okay, so I hope you understand what is the cytosol and what is the difference between cytoplasm and cytosol. Cytoplasm, it means that all the organelles are found within the cell membrane except to nucleus, that is called cytoplasm. Okay, and cytosol is having a liquid matrix, and that will found around the outer skirt of the organelles inside this cell membrane, that is the cytosol, and that will be in the form of organic molecules, salt or water. So here we can say that the same thing are told here in the form of the sentence. Cytosol is thus a liquid matrix around the cell organelles. In prokaryote, most of the chemical reaction of metabolism take place in the cytosol, while a few take place in the membrane or in the periplasmic space. Okay? And in eukaryote, while the many metabolic pathway still occur in the cytosol, others contained within the organelle. So in whole cell, I can say that this is the one of the basic site for the most of the chemical reaction inside the cell. So our answer is the cytosol. Let's move for the another question. Another question is saying that in ninth, which cell organelle is the site of chemical activity in cell, perhaps overall half of the cell metabolism? So our answer will be mitochondria. So here we can see the structure of the mitochondria. Mitochondria looks like that. These are the different structure. There will be the intermembrane and ribosome, DNA. This is your genetic material, F0 F1 complex, and this is crest. This is the crest, and here will be the inner membrane, outer membrane, and this is the matrix. So this is the actually the different part of the mitochondria. Now the main function of this is it is actually the site of chemical activity in cell. Perhaps more than half of the cell metabolism or occur on that. That is actually our answer is the C, mitochondria. Let's move for the another question. Another question is saying that photorespiration is high in. So photorespiration is high in rice, because rice is a C3 plant, and maize, sugarcane, and pineapple, these are the C4 and CAM. So photorespiration are absent in C4 plant as well as the CAM plant, but is present in the C3 plant, and our answer is the C, rice. Let's move for some more explanation about this photorespiration. It is actually also known as the oxidative photosynthetic carbon cycle, or C2 photosynthesis, sometimes we can say it. In this process, the plant metabolism, where the enzyme Rubisco oxygenate RUBP, and because of this enzyme Rubisco, and that will oxygenate this ribulose biphosphate, and that will cause the some of the energy that will produce during the photosynthesis by the photosynthesis will be wasted. That's why this is a destructive or waste process, and that's why the C3 plant have the less productivity as compared to the C4 plant, because most of the energy that they lost in the form of photorespiration, because energy losses will occur in this process. If you see where this actually the photorespiration occur, so actually this photorespiration will occur in chloroplast, peroxisome, and mitochondria. Many times it has been asked that in which cell organelles photorespiration occur. So your answer will be this: chloroplast, peroxisome, and mitochondria. If suppose these three organelles are available in that answer, so that will be your right answer. So here you can see this Rubisco this will cause the oxidation of ribulose biphosphatase, and that will lead to the oxidation or wastage of the energy that will be oxidized. That's why the energy that we produce during the photosynthesis will be wasted in the form of photorespiration. So I hope this explanation will be very useful to you. You are able to know what are the different physiology questions and with their explanation. I tried my best. If suppose some mistake is there, so you can comment in my box. That will be definitely will be helpful for me to improve my video in the future. And I hope this video will be very useful to you, and please share this video in your friends, and please don't forget to like also, because your like and comment will make definitely improvement in my video in future. Thank you.